Welcome to this explainer. Today we are looking at something absolutely fascinating, what is essentially the ultimate biological obstacle course, human reproduction. For literally millions of years, natural selection has been designing this incredibly rigorous, almost impossibly difficult journey for a sperm to reach an egg. It's a completely flawless filtering system, and it's designed by nature to ensure that only the absolute healthiest, most structurally sound genetic material is ever passed on to the next generation. But we have to ask ourselves, what happens when human biology struggles? What happens when we actually have to step in and try to recreate that intense microscopic obstacle course inside a laboratory? Well, today we are diving deep into a really comprehensive, newly published 2025 review on assisted reproductive technology, or ART, from the Journal of Assisted Reproduction and Genetics. We're going to explore exactly how modern science is stepping in, right now, to beat the odds and engineer the quest for the perfect sperm.
Okay, let's dive into this because the stakes right now are incredibly high. The source data in this review reveals a staggering, frankly alarming reality. Over the last 45 years, specifically between 1973 and 2018, global sperm concentration has plummeted by a massive 60.3%, and it's not stopping. It is actively decreasing at a rate of roughly 0.87 million per milliliter every single year. Just let that sink in for a second. Because of this massive global decline, male factors now actually contribute to a full 50% of all infertility cases. That's half. This reality makes advanced treatments like in vitro fertilization, and more specifically, intracytoplasmic sperm injection, which we generally call ICSI, an absolute necessity for millions of couples worldwide who are just trying to start a family.
To really understand the sheer scale of the challenge that scientists face today, we have to respect the brutal but totally necessary reality of natural selection. In a natural conception scenario, the physiological selection process is so unbelievably unforgiving that only 1 in 10,000 sperm actually ever reaches the oocyte or the egg. Think about that ratio. The female reproductive tract acts as this incredibly harsh, complex environmental filter. However, in the lab, especially during an ICSI procedure where an embryologist is manually picking up and injecting a single sperm directly into an egg, we are completely bypassing that natural filter. And when we bypass nature's rigorous selection process, we dramatically increase the risk of accidentally using an abnormal sperm. That is a massive problem because it can lead directly to embryonic developmental anomalies.
So, how are scientists solving this? We'll be following a clear 6-part roadmap today. First, global infertility and natural selection. Second, the limits of traditional methods. Third, immotile but viable sperm. Fourth, microscopic and structural examinations. Fifth, functional selection and microchips. And finally, the future of IVF technologies.
Let's start with Section 1, Global Infertility and Natural Selection.
So, the crucial point is understanding exactly what it is we are trying to filter out when we recreate this obstacle course. The real hidden enemy preventing live births today is something called sperm DNA fragmentation, or SDF. Essentially, this refers to the actual physical breaking or damage to the very genetic material housed inside the sperm itself. The 2025 review emphasizes over and over that sperm quality defects, and particularly this impaired DNA integrity, are completely central to the limitations we currently see in ART success rates. High SDF directly correlates with diminished success in the laboratory, much lower fertilization rates, and it even poses severe epigenetic risks. That means it can actually negatively affect how the offspring's genes are expressed down the line. It's not just about getting pregnant, it's about the health of the future child. We absolutely have to find a way to leave these damaged sperm behind.
Before we start looking at the actual lab methods, I really want to highlight the scientific rigor backing the information we're exploring today. The researchers behind this review didn't just casually skim a handful of papers. They started with a massive pool of 4,345 original records. Then, through a highly rigorous screening process that applied the strict NHLBI-NIH quality assessment guidelines, they meticulously filtered out thousands of studies. They narrowed it down to arrive at just 42 of the absolute highest quality clinical trials available. These 42 rigorously vetted studies form the solid foundation of everything we're breaking down in this explainer today.
Moving on to Section 2, the old guard and the limits of traditional laboratory methods.
For years, clinics all over the world have primarily relied on 2 traditional methods to isolate sperm: swim-up and density gradient centrifugation, or DGC. Now, swim-up is exactly what it sounds like. You place the semen sample under a culture medium, and you basically rely on the motile, healthy sperm to actively swim against gravity up into a clean upper layer. It's a very gentle process with minimal damage, which is great, but it has a notoriously low recovery rate. That makes it pretty much useless for patients who already have low sperm counts. DGC, on the other hand, is the most common standard practice today. It works by forcing the sperm through these discontinuous density layers in a spinning centrifuge. The idea is that the healthy, highly motile sperm are dense enough to make it all the way to the bottom, while the cellular debris and the abnormal sperm get trapped in the upper layers. But here is the major catch that researchers are pointing out. The DGC process itself can actually stress the sperm out. It can increase what we call reactive oxygen species, or ROS. And most importantly, neither of these visual traditional methods can easily identify if the actual DNA inside the sperm is intact or fragmented. They just look at how it swims or sinks.
This brings us to Section 3, immotile but viable sperm, or how embryologists go about finding life when nothing is moving.
So imagine you're an embryologist looking through a microscope and none of the sperm are moving. How do you know which ones are actually alive? One absolutely fascinating solution to this challenge is the hypo-osmotic swelling test, commonly called HOST. This test cleverly relies on the functional integrity of the sperm's outer membrane. Out in nature, a sperm has to withstand really intense osmotic pressure changes as it travels from the male epididymis all the way to the female reproductive tract. In the lab, embryologists mimic a pressure change by placing the sample in a hypo-osmotic solution. If the sperm is viable and alive, its membrane will immediately react to that pressure change by absorbing fluid. This causes the tail to characteristically curl or swell up. Dead sperm? They simply don't react at all. It's a brilliantly simple way to spot a living sperm without having to use any toxic chemical stains. I will say, though, as the researchers point out, having a curled-up tail definitely makes that sperm quite a bit harder to physically catch with a microscopic needle for injection.
Another truly incredible high-tech technique for this exact scenario is laser-assisted immotile sperm selection, or LIAS. Here is exactly how this step-by-step procedure works in practice. First, under a high-magnification microscope, the embryologist searches for an immotile sperm that at least looks morphologically normal on the outside. Next, they fire an incredibly precise targeted laser pulse right at the very tip of that sperm's tail. If that sperm is alive, it physically flinches. The tail will immediately curl or bend in a rapid reaction to the laser-induced influx of the surrounding medium. It's literally a reflex test for a single cell. The real beauty of this method is the precision. By firing the laser at the very furthest tip of the tail, the energy is kept safely away from the head of the sperm, theoretically protecting that precious DNA cargo inside from any thermal damage. Let's dive into Section 4, microscopic and structural examinations. Enter a technique called MSOE, which ultimately leads to a clinical procedure known as IMSI. This technique represents just sheer microscopic power. Just to give you a comparison, while a standard ICSI procedure magnifies a sperm about 400 times, IMSI combines optical microscopy with advanced digital software to blow that image up to over 6,000 times magnification. It's wild. At this massive, granular scale, an embryologist isn't just looking at the general shape of the sperm anymore. They are meticulously inspecting its specific internal organelles. They're checking the acrosome, the post-acrosomal region, the mitochondria in the midpiece, and most importantly, the nucleus where the DNA lives. They can actually spot microscopic craters or tiny vacuoles in the sperm head that would normally be completely invisible under standard microscopes. Now, because it requires highly specialized, expensive equipment and extensively trained embryologists, it isn't standard practice everywhere just yet. But the data clearly shows it is a remarkably powerful tool, especially for older couples or those who are unfortunately facing repeated implantation failures.
And this brilliantly illustrates another way we can use light to our advantage: polarized light microscopy. What's so cool about this method is that it doesn't rely on adding any harsh chemical dyes to the sample. Instead, it relies entirely on the intrinsic natural optical properties of a perfectly mature sperm. You see, in a healthy mature sperm, the highly condensed chromatin packed tight in the nucleus, along with the intact acrosomes, exhibit a phenomenon called birefringence. Basically, when you view them under polarized light, these specific healthy structural elements literally glow. On the flip side, sperm with abnormal morphology or poor motility usually show these atypical dull or dark patterns. It's an elegant solution that allows the embryologist to visually confirm the internal maturity of the sperm head just by observing how its structures bend the light.
Now we reach Section 5, functional selection and microchip innovations, the ultimate obstacle course. So far, we've looked at visual tricks, but researchers have also designed 3 distinct biochemical traps to test a sperm's actual functional maturity. First in our table is zeta potential. Mature sperm naturally develop a negative electrical surface charge, specifically right around -16 to 20 millivolts. By intentionally applying a positive charge to a standard plastic tube, the mature, negatively charged sperm are magnetically attracted and literally stick to the walls, completely leaving the immature ones behind. Second is electrophoresis, which scales this electrical concept up by using active electric fields to isolate the functionally competent sperm. The big advantage here is that it significantly reduces DNA fragmentation and limits that oxidative stress we talked about earlier. Finally, we have HA binding. Hyaluronic acid, or HA, is a substance naturally found in the dense cellular matrix that surrounds a human egg in the body. Only fully mature sperm have developed the highly specific glycoprotein receptors needed to actually bind to and digest this acid. So, by coating a petri dish in HA, embryologists can literally watch to see which sperm managed to bind to the surface, decisively proving they possess the biological keys required for natural fertilization.
Now, what's really interesting about this slide is a technique called MAX, or magnetic activated cell sorting. If I had to describe it simply, this is essentially a way to hunt down zombie sperm. These are sperm that are swimming around and might even look totally normal under a microscope, but functionally, they have actually already begun the irreversible process of cellular death known as apoptosis. When a sperm starts dying, a specific molecule called phosphodiesterine flips and pushes through to the outer layer of the cell membrane. To catch them, scientists introduce tiny, specialized magnetic beads into the sample. These beads are coated in a protein called annexin V, which acts exactly like a homing missile for that specific death signal molecule. The magnetic beads bind exclusively to the dying zombie sperm. Then, the embryologist simply turns on a magnetic field, which quite literally drags all the dying, damaged cells out of the way, leaving behind a pristine, untouched population of healthy sperm ready for the ICSI procedure. But perhaps the absolute most exciting advancement detailed in this 2025 review is microfluidic sperm sorting. Truly, this is the closest thing science has ever built to a lab-made biological obstacle course. Rather than violently spinning the sperm in a potentially damaging centrifuge like the old methods, researchers now use highly advanced microfluidic chips. These incredibly small devices feature complex, microscopic fluid channels that are engineered to perfectly mimic the natural physiological environment and the complex fluid dynamics of the female reproductive tract. The sperm are placed at the start and forced to actively swim their way through these intricate micromazes. Only the absolute strongest, most structurally sound, and highly motile sperm can successfully navigate the fluid resistance to reach the collection chamber at the end. The studies analyzed in the review consistently show that this gentle yet intensely rigorous method yields sperm with vastly superior DNA integrity compared to any of the traditional methods. Let's move to and see how this builds into our final section, Section 6, the future of IVF technologies and precision ART. If there is one main takeaway from this comprehensive 2025 review, it's that, right now, there is no single, undisputed winner. There is no one magic laboratory technique that works perfectly for every single patient who walks through the clinic doors. Traditional methods like swim-up and DGC are absolutely still the mainstream choices because they are stable, well understood, and broadly applicable for many baseline cases. However, for patients dealing with severe infertility, repeated failures, or known high DNA damage, the advanced techniques we discussed today, like microfluidics and ICSI, are absolute game changers. The true future of this field lies in combining these incredible tools with emerging, cutting-edge technologies like artificial intelligence and single-cell omics, which is the ability to study the molecular makeup of individual cells in unprecedented detail. By analyzing massive, complex datasets, AI will very soon be able to help embryologists look at a patient's specific profile and choose the exact right combination of these advanced sorting methods. We are moving toward an era of highly personalized, precision treatments based entirely on a patient's unique biological needs.
Which leaves us with a rather profound question as we wrap up this explainer. As our microfluidic chips get more and more complex, and our microscopes and lasers get even more precise, will human technology ever be able to perfectly replicate nature's millennia-old, flawless filtering system inside the lab? We are undeniably getting closer every single day, bringing vital new hope to millions of families around the world who are struggling to conceive. But the ultimate limits of human engineering, when stacked up against the ancient, rigorous designs of natural selection, well, that remains to be seen. Thank you so much for joining me on this incredible journey through the microscopic world.